Hello, my name is Joanne Nazario, and we will be talking about the cell membrane. Topics of discussion for today are the cell membrane structure, cell membrane function, different classes of membrane proteins, different types of diffusion and transport, and energy. All cells have plasma membranes. Although they can be different, all cells have a membrane. A lipid bilayer is two opposed layers of lipids, structuralized tail to tail. The tails of the lipids are hydrophobic because of their hydrocarbons. The heads are hydrophilic because they contain a positive charge. The membrane is overall hydrophobic and water repellent for protection, but contain both hydrophobic and hydrophilic characteristics, meaning they are amphipathic. These lipid structures exist in a watery environment, and the membrane must be insoluble. When exposed to water, the orientation of the membrane contains an interior projection of hydrophobic tails and an outer orientation of the hydrophilic heads. The hydrophobic components of the cell membrane are content with the membrane excluding from water. Oils must be separate from water. The lipids are stuck in the membrane, but they can float forward, backwards, side to side. This is known as lateral diffusion and is noted as having 2D fluidity because there is no up or down 3D movements. There are also various membrane proteins that are found within the cell membrane structure that are used to aid cellular activity. The membrane is a thin fatty membrane that protects the cell components from outer environments. All living cells must be organized and compartmentalized. Membranes are selective and determine what nutrients, waste, or food can enter and leave the cell. Although the membrane is semi-permeable, that does not mean that anything and everything can enter and leave. The lipids compose the barrier, while the membrane proteins can be the doors or portals. The two types of membrane proteins discussed are pumps and channels. Pumps are regulated in a manner of controlling what's being moved from lower concentration to higher concentration using energy. Channels can be passive in what moves in and out of the cell. Another function of the cell membrane is transport. The cell membrane uses membrane proteins in order to aid the regulation of entrance and exit of molecules into and out of the cell. Cells also have to be fluid. So in order to maintain that fluidity, there is an orientation of lipids that must be maintained. The fluidness of a membrane is known to be as fluid as oil. Looser fluidity is characterized like water. Jello is an example of tighter fluidity. Membrane fluidity can be regulated by compacting lipids to determine dense or loose membranes. The double bonds and hydrocarbon tails can determine the denseness or fluidity found within the membranes as well. Longer hydrocarbon tails have more double bonds, meaning more kinks, which mean less proper orientation for density. Shorter hydrocarbon tails do not contain double bonds and allow for a better regulation and orientation. Cholesterol contributes to the density and allows for the filling between the kinks in the longer hydrocarbon tails. Cholesterol fill the gaps and allow for less fluidity. Less fluidity allows for stiff, less mobile membranes. The lipid bilayer is known as asymmetrical because of the distinct characteristics and functions of the inner and outer phospholipid layers. The smooth ER synthesizes lipids and exert vesicles of lipid bilayers that merge and fuse to membranes and regulate the correct face. Membrane domains are specialized regions of collected membrane proteins. Many of the outward-facing lipids are glycolipids. These are lipids that have sugars, oligosaccharides, linked to the heads of the lipids. These sugars allow for another protective layer. Cells are living organisms that are exchanging molecules within its environment. The hydrocarbon tails are hydrophobic and make it very specific in regulated membrane transportation. It is known that the hydrophilic molecules are very important for life and have to be able to move through the hydrophobic lipid areas. Unique transport proteins are used to allow for charged hydrophilic molecules in and out of the cell. There are various transport proteins that are specialized for individual molecules. Transporters are proteins with moving parts that allow for movement and molecules through the membrane by changing shape. It is rare for these proteins to move molecules in and out at the same time. Transport proteins often move molecules against their gradient. This requires energy and is known as active transport. A hydrophilic hole that allows the passive transport of ions without the use of energy is known as an ion channel. Ion transport is highly critical for cells' membrane concentration. The most critical ions that have controlled concentrations in transport are sodium, potassium, chlorine, and proton ions. Sodium ions concentrations are found extracellular and potassium concentrations are found intracellular. Potassium is positively charged and chlorine balances out that charge occasionally. Ions are charged and extremely hydrophilic. This creates problems with ion diffusion. Transporter proteins and channels are regulated by the cell in order to allow for ion regulation and movement. This regulation must also have a regulated time span for transport. There are small nonpolar molecules that can easily diffuse through the membrane without help. Those include oxygen, CO2, and N2. There are also small uncharged polar molecules that can diffuse into the cells, such as water and ethanol. There are also large uncharged polar molecules that cannot diffuse efficiently or fast enough without the use of a transporter or a channel. These include amino acids, glucose, and nucleotides. The last group is all charged ions. No matter what size they are too hydrophilic and cannot diffuse without the help of membrane proteins. Those include sodium, potassium, calcium, chlorine, and many more. All membranes have transport proteins that are specialized and associated with specific molecules. The size and charge are the reason for channel specificity. There are a few types of membrane proteins and various types to classify them. If we're going to classify by embedment, we are going to classify them as either integral proteins, peripheral proteins, or anchor proteins. Transport proteins are the active protein that moves molecules into the cell. Anchor proteins link intracellular actin with extracellular matrix. Enzymes are membrane proteins that catalyze responses to signals from cell interior and exterior environments. Proteins that are embedded are hydrophobic. To exist in the hydrophobic environment of the membrane, they must be integral proteins. In order to remove and liberate these proteins, detergents are used. Any portion of proteins that touches the tails or inner portion of the membrane are known to have hydrophobicity. There can also be proteins that associate with a second protein stuck to the lipids, or a secondary hydrophobic protein. And these are known as peripheral proteins. So there are different types of diffusion and transport. Simple diffusion is the movement of molecules moving from high concentration to low concentration without any help. Facilitated diffusion or passive transport is the same concept of high concentration to low concentration, but it needs the help of protein channels because it cannot enter easily through the membrane. Active transport is the movement of molecules from low concentration to high concentration. Moving against the concentration gradient requires energy in protein pumps because it is not naturally done. Different organelles have different functions and have a need for different concentrations of molecules, and this leads to the variety of channel and pumps. The electrochemical gradient can either oppose the molecule's concentration gradient, allowing for opposing directions. There is the use of a coupled transporter that uses one molecule to drive the power of the other using the natural gradient to obtain that energy. The use of the energy of one molecule natural tendencies is harnessed, and that energy will propel the unnatural low to high concentration movement of the other molecule.